Okay, so let's pull up the share screen. All right. So let's continue our journey about DNA replication. So what we're going to do is talk about the basics of DNA replication. Again, as per the usual is what I do. This is an active link to your the textbook for this section. Okay. So what we need to do is understand how DNA structure actually reveals how DNA replication occurs. And then we'll talk about the different models of DNA replication, whether that's conservative, semi-conservative, or dispersive. And then how the Melson and Stahl experiments correspond with that. Okay, so remember from the Watson and Crick paper, they had this little sentence at the very end that said, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And so remember that the structure of DNA is this double helix, as is shown in this image here, where we have the sugar phosphate backbones running in anti-parallel structures directions from each other, and then the rungs of our ladder are the bases that are in the interior of the double helix. And so maybe take a second and think about, how does this structure suggest how DNA is copied? So if we untwisted this double helix, we would have two single strands of these DNA molecules with these bases that are sticking out that we can actually use as a template to create a new DNA molecule. And so that idea of the semi-conservative copying goes like this, that our original data, our original DNA is found in a double helix wrapped around each other. We untangle that and melt it so that we break those hydrogen bonds between the bases that are keeping those rungs of the ladder together. And that's what we actually find out seeing, is that it is a semi-conservative model of DNA replication. The alternatives were this conservative model where DNA replication occurs and we have a completely new double helix and our old double helix. And then the dispersive model says that the new stuff and the old stuff is kind of dispersed in between the same strand of DNA. And so that was the question, is how does it actually replicate? And because what we knew at the time was that about half of the genetic material was conserved and half of it was new stuff. And so they were like, okay, well, how does this actually work? So 5 years after Watson and Crick's study was about the structure of DNA was published, this other study was published that showed how DNA actually replicates using a semi-conservative replication model. So what these two fellers did was that they grew a bunch of bacteria, E. coli, on media that was spiked with heavy nitrogen. So remember we talked about, like, well, who cares about isotopes? And I said, if you care about DNA replication, then you care about isotopes. So most of the time, most nitrogen atoms have 7 protons and 7 neutrons. But some of them have an extra proton. So they're actually have a molecular weight of 15. And so they're heavy nitrogen, or the isotope we call is N15, right? So we find nitrogen in the bases, but not in the sugar or the phosphate. And so what these two researchers did was they took their bacteria they grew grew some on the heavy nitrogen media and then some on the regular nitrogen media. And then they put them all, some of the equal parts into another container containing the nitrogen 14, the regular nitrogen. And so what they found, the reason that they use this heavy nitrogen is because you can put it into a little tube and you put it in the centrifuge and you just spin the heck out of it at I think they were using like 32,000 rotations per minute. So that's like stupid fast, right? And what happens because of the DNA that has the heavy nitrogen is because it's more dense, is going to fall out at a lower level than the regular light nitrogen. And so, so at generation 0, they just plop some of these dudes that they've only ever had N15, and they're finding out, yeah, sure as heck, their band is coming out as expected. And so then after one generation of them growing on this new media, they're finding that the band is actually coming out a little bit lighter. And there's just one single band that's kind of somewhere in between nitrogen 14, nitrogen 15. And so they're like, okay, so that suggests that 50% of the genetic material in these bacteria have heavy carbon or heavy nitrogen, and half of them have regular nitrogen. That's what we expected. So, and then for another generation, they grew them again in just regular nitrogen. And then we started to see the separation of two bands, where we see some of these bands have that same density as from generation 1, indicating that half of that has been conserved and that half of it has been created new. And so then we took that another generation, and we can see as we go through all these generations, the proportion of heavy nitrogen is falling away at a predictable rate. If we're just doing half of what was in the previous generation. And so that is the evidence for this semi-conservative replication. As it's reproducing, it's keeping some of that heavy stuff, but it's also producing new nitrogen, and it's decaying away at a predictable rate. And so that's what this semi-conservative model is. So if we pretend that this little purple sugar phosphate backbone has been labeled with a heavy isotope, then we start to see, okay, so now we have one of those strands goes to each of the daughter DNA strands, and half of that double helix is new stuff that's produced with a regular nitrogen. And then in our second generation, we see that there's one strand that's still labeled with a heavy isotope that was way back in our parent, right? So like we're seeing this decay, 50% of these strands has the heavy isotope. And then we are seeing that, okay, there's 1, 2, 3, 4, 5, 6, 7, 8. Only 2 of those 8 have the heavy stuff. And so we can see the decay. And so these guys grew the bacteria for 14 generations, and you did see this very predictable pattern that you would see with semi-conservative DNA replication. Okay, that is it for this section. So we'll pick up with how prokaryotes and eukaryotes do DNA replication, how it's the same and how it's different in a bit.